So our next speaker is Katarzyna Kanarek. I hope I pronounce it correct. Not so much, I apologize. From Dor Solomon's lab. Thank you.
Hello everyone, I'm Katarzyna Kanarek from Dor Solomon's lab. I'm doing PhD there. Today I would like to share with you some fascinating but also slightly terrifying findings from our lab regarding the pathogenic potential of environmental vibrios lurking in Israeli coastal waters. But before I start, whomever here is above 40 likes drinking wine and enjoys swimming in the sea during summer, I would maybe advise to change the summer plans, because there is a chance you will meet the flesh-eating bacteria, Vibrio vulnificus. Every year we hear about infections caused by this bacteria. They tend to infect individuals I described earlier, but they also infect people with pre-existing conditions like liver disease, weakened immune system, or diabetes. They are highly virulent and they can cause infections, which mortality rate reaches up to 50%. Actually, we had our own outbreak of Vibrio vulnificus in the 90s, which originated from 2 fish ponds. What happened was it was very hot, so fish sellers decided to not wrap fish in ice, instead just selling them alive in water. So once they went back to their own old habits of selling fish in ice, also the Vibrio infections dropped. So who are these little rascals? Vibrios are gram-negative rod-shaped bacteria. They are naturally in marine environments. The Vibrio vulnificus that I previously mentioned, but also Vibrio parahemolyticus, Vibrio cholera, Vibrio genolyticus, these are human pathogens and they can cause serious illness. What is also important to mention here, Vibrios very easily share their genetic material between each other, and this results in the emerging of the new pathogenic strains. So why am I scaring you like this? It seems that the global warming is the cause of all our problems these days, but unfortunately, because of the global warming, also the temperature of water in oceans rises, which gives perfect conditions for vibrios to thrive. And the more Vibrio bacteria in the sea, the more Vibrio infections we observe across the globe. So the National Public Health Laboratory in Jerusalem each year collects data from all the labs in Israel and summarizes them. So in the 2020 summary, which they released, we can see that there is an increase in Vibrio infections. There's an increase in Vibrio genolyticus infection, but more importantly, increase in Vibrio cholera infections. So these strains here, they don't produce CTX toxins, so they are not responsible for cholera outbreaks. However, they are still pathogens of humans and they can cause serious illness. So here, I just wanted to show you that Vibrio cholera was predominantly found in blood and ear samples, while genolyticus from ear. So these summaries, these studies are important. However, we believe that they may lack some in-depth analysis of the actual vibrios that cause those infections. They also are limited to Vibrio pathogens that are human pathogens, and we know that Vibrios can cause damage also to, they can infect marine life, which can cause damage for aquaculture or even for our corals, such the ones in Elat. Another thing is that here we are missing all the data from vibriosis, which are the Vibrio infections collectively called. We are missing all the data from vibriosis which are passing on their own, untreated. So our lab responds with a research that aims to characterize the virulence potential of environmental vibrios, but also to test their antibiotic resistance, to to see what's going on out there, because the more we understand the vibrio populations, especially in our waters around us, the better prepared I think we can be to fight these infections or to predict what will happen next. So what we did, we collected 3 sample waters from 3 different places in Israel during summer 2023. We used special TCBS plate. This medium is designed to isolate vibrios. So we use this to isolate only vibrios from our water, but we also checked the morphology on different plates that we use in our lab. So once we had our vibrios isolated, we sent them for sequencing. And what we found out was that actually we were able to isolate a quite variety of vibrio species population. We even isolated a novel vibrio species, which we named after our friend, the late Professor Guido Sessa from Life Sciences. But what I want to show you here is that we were actually able to isolate quite a number of pathogens that can cause public health, that can be a public health risk, like Vibrio allergenolyticus, Vibrio parahemolyticus, but we also isolated Vibrio vulnificus, this flesh-eating bacteria, unfortunately the one who is causing this disease. So once we had our sequences, we could look into them and try to find some indicators of pathogenicity. So we decided to look at type 3 secretion system proteins, but also some secreted toxins that these bacteria have. So in our pool of bacteria, we found out that 39% of them had type 3 secretion system 1 or more, which is very high. So type 3 secretion system is a secretion system that allows pathogens to secrete anti-eukaryotic proteins outside to the host cytoplasm directly from this needle-like structure. So next we wanted to look at the toxins they secrete. So here I just name a few. We found a lot of them. But among the toxins that we found out, they were predicted to target membranes of eukaryotes. They were predicted to interact with the cellular processes directly. We found TC toxin. We found pure AB toxin that are known to cause mortality in shrimps in aqua farms. Lots of different toxins, but that diversity suggests how complicated host pathogen interactions are. So once we knew they carry some bad stuff with them, we wanted to test it. We wanted to establish their virulence potential against the BMDMs. These are bone marrow-derived macrophages. So we are very lucky to collaborate with Professor Moti Gerlitz's lab, and his very talented PhD student Hadar Cohen performed all the infection experiments for us. So here is this is just an example of this infection, how it looks like. On the left side, we have very neutral Vibrio fortis, and on the right side, we have quite aggressive Vibrio parahemolyticus. So what she did, after checking all the bacteria we isolated, 61% of them were detrimental to macrophages, which is quite scary because these were just bacteria we randomly isolated from waters. So next we wanted to answer the questions about the antibiotic resistance. So we selected 4 antibiotics that are commonly used in treating of vibriosis. We acquired strips with antibiotics commercially available from Biomerie. And these were this is the susceptibility pattern that we have. So first I want to mention that at the very bottom we have Vibrio parahemolyticus RIMD. This is a clinical strain. So unfortunately, not surprising, it is quite sensitive, quite resistant to all of them. But what we can see from here, what I want you to remember is that more than 50% of them were resistant to ciprofloxacin and azithromycin. 35% of them were resistant to the rest, the ceftriaxone and doxycycline. So it's quite scary. Let me tell you that it's quite scary. Although I see some light in the tunnel, first thing that I see is that we didn't find any bacteria that is resistant to all of them, which is good. Another thing is that here at the bottom, we have another Parahemolyticus. Despite being more resistant to doxycycline, it is more sensitive to other ones, so that's good. So take-home notes. Take-home notes, don't swim in the sea during summer, especially if you have a freshly made tattoo or any wounds. Why? Because we found a lot of we found a lot of anti-eukaryotic. No, we found a lot of very, very nasty stuff swimming there, especially flesh-eating bacteria. And we just see that there is need to monitor our waters, but also we see the need to actually test vibrios from patients to establish, to look closer into them. So at the end, I would like to thank so much for TCCP for all the grants they enriched me with, scholarship and also the travel grant. Special thanks to Kinga for helping me with testing antibiotics. And also thanks to Hadar, who did all the infection experiments. And thank you so much for listening.